This screencast covers the topics of the nucleus and DNA replication, which you will find in Chapter 3 of your textbook. The learning objectives of this screencast are as follows. Describe the nucleus in detail, briefly describe the function of the nucleus, explain how DNA is organized in dividing and non-dividing cells, describe the function of the nucleolus, describe the process of replication including the location and the enzymes involved, explain why DNA replication is referred to as semi-conservative replication. Let's now talk about the nucleus. The nucleus is centrally located in a cell, usually, and it typically takes on the overall shape of the cell in general. Most cells have at least one nucleus, at least initially. Some cells may have multiple nuclei when they are mature, or they are what we call multinucleated cells. An example would be skeletal muscle fibers or cells. Some cells, when they're mature, have no nuclei at all, such as the red blood cells. The nucleus is often referred to as the control center of the cell, and that's because it is where the DNA or genetic material of the cell is located. Recall that the DNA contains the code or the recipe for all of the proteins that a cell makes. The nucleus itself is composed of three regions: the nuclear envelope, the nucleoplasm, where the chromatin is located, and the nucleolus. The nuclear envelope separates the nucleus from the cytoplasm. It is an effective barrier controlling the flow of materials into and out of the nucleus. The nuclear envelope is composed of two membranes, each being very similar in structure to the plasma membrane. The nuclear envelope contains nuclear pores, which allow molecules to move between the nucleus and cytoplasm. The nuclear pores are formed by the fusing of the inner and outer layers of the nuclear envelope. Filling the interior of the nucleus is the nucleoplasm. The nucleoplasm is very similar in composition to the cytosol, with areas or regions of chromatin usually dispersed within. What is chromatin anyway? It is the form in which DNA exists in a non-dividing cell. And cells spend most of their life cycle not dividing. So usually DNA is in the form of chromatin. Chromatin consists of DNA and proteins called histones. So as you know, DNA is a double helix. It's a very thin but very long molecule. This means that it is easily broken or fragmented. To protect the DNA from fracture, it is wound around proteins called histones. It is the accumulation of the histones with their wrapped DNA that forms chromatin. Now, chromatin is a very unorganized form of DNA. It would be very difficult to segregate the DNA in this form during mitosis and cell division. Therefore, in preparation for cell division and during cell division, chromatin is condensed into chromosomes. Although only one chromosome is shown here, the human body contains 46 chromosomes. Found within the nucleus are one or more dense tangles of chromatin called nucleoli. Nucleolus is singular. The components of the ribosomes are synthesized at the nucleolus. They are then transported out of the nucleus through the nuclear pores, where they are assembled in the cytoplasm. The ribosomes are then attached to the rough endoplasmic reticulum, or they remain free in the cytoplasm. Now let's discuss the duplication of DNA, which is referred to as DNA replication. If you recall from our cell life cycle and cell division lab, we learned that in the G1 or GAP1 subphase of interphase, a chemical signal is received by the cell indicating that it is time for the cell to undergo cell division. In order for the two new cells that are going to be formed from the original parent cell, it is necessary for all the DNA to be duplicated so that the two new cells will have DNA identical to the original parent cell. So during the synthesis or S subphase of interphase, the DNA will be replicated. The very first activity that occurs during replication is an enzyme called DNA helicase unwraps the two strands from one another. Remember that DNA is double-stranded with nucleotide bases connected to one another by hydrogen bonds. So this helicase breaks the hydrogen bonds and pulls the strands away from one another. And where they're being pulled apart, that forms a replication fork as shown here. As the strands are pulled apart, you get a sort of a bubble in the strand of chromatin. You can imagine what that would look like. Here you have your strand here, and then all of a sudden, boom, here is a little bubble in that strand. Please note that you're going to have these replication bubbles on several locations on a chromatin thread. Although it's only being shown that it's in one location, you can imagine that it's occurring at several locations on a given chromatin thread. And it's going to be occurring on different chromatin strands at the same time until replication is completed. With these two strands now pulled apart, a second enzyme is going to come in called DNA polymerase. And DNA polymerase is going to use our old strands as templates, and it's going to come in and assemble complementary nucleotide bases on the new strand here, and as you can see here. Now notice that these two new strands are being produced in opposite directions. The strand that's being formed in the direction where the DNA molecule is being pulled apart is referred to as the leading strand, and the strand that's being formed away from where the DNA is being pulled apart is called the lagging strand. DNA polymerase can only work in one direction with respect to the complementary bases, and so that's why the leading strand and the lagging strand are formed in opposite directions. So eventually the DNA polymerase molecules assemble complementary new strands along the entire length of the original DNA molecule. resulting in 2 new daughter DNA molecules. Notice that each new DNA molecule consists of one of the original strands, shown here in blue, and one of the new nucleotide strands, shown in red. Because each new synthesized DNA molecule consists of one old template strand and one new nucleotide strand, DNA replication is also referred to as semi-conservative replication. With the DNA now fully replicated, histone proteins which were synthesized at ribosomes in the cytoplasm are imported into the nucleus, and the DNA is wrapped around them to form new chromatin. The cell then moves into the gap 2 phase and makes final preparation for cell division. Often, replication results in damage or errors in the DNA. The cell cycle will remain at the G2M checkpoint until any DNA damage is repaired. After that, the cell will move on to the mitotic phase and complete mitosis and cell division. Now let's review the learning objectives of the screencast, describe the nucleus in detail, briefly describe the function of the nucleus, explain how DNA is organized in dividing and non-dividing cells, describe the function of the nucleolus, describe the process of replication, including the location and the enzymes involved, explain why DNA replication is referred to as semi-conservative replication.